Okay, 7 o'clock. We still have people trickling in, but maybe we will get started. Looks like we're live on Facebook as well. So thanks everyone for tuning in to tonight's webinar. We weren't actually going to host a webinar in July, but fortunately for us, Dr. Skinner recently published a paper and the timing has worked out exceptionally well. And given the state of steelhead in BC, we figured we should bring it to you. And fortunately for us, it worked out in his calendar. So a number of you probably been in on the webinars in the past. The layout here is a presentation around 45 minutes, and then we'll do question and answer for 15 to 30 minutes. You can put your questions in the Q&A box at the bottom, or on Facebook Live, you can put them in the comment section, and we will put them all together and then ask them at the end of the presentation. The objective for these webinars is to help educate you so that you have a better understanding when you're advocating with your elected officials and when you're talking to your friends who care about fish and wildlife. So just want to keep it top of mind that, you know, knowing about this is really good, but passing that knowledge on to other people is even better. And that is how we will take care of our fish, wildlife, and habitat resources. A little bit around the question and answer piece. Just want to make sure everyone knows that Dr. Skinner is not a policy or decision maker. So if you feel your blood pressure rising, feel free to engage your elected officials on those kind of issues. Dr. Skinner here is he's here in the context of being a scientist and a researcher. He's certainly up on a lot of the questions, and he has said that he can talk about a little bit about policy issues and hatchery related issues, but just keep in mind, he is not the person who makes all the decisions. He's here to share his wealth of knowledge and the findings from his science. So I'd like to give a quick intro to Dr. Skinner. So Dr. Michael Skinner is a professor in the School of Biological Sciences at Washington State University in Pullman, Washington. He did his Bachelor of Science in Chemistry at Reed College in Portland, Oregon, his Ph.D. in Biochemistry and Chemistry at Washington State University, and a postdoctoral fellowship at the C.H. Best Institute at the University of Toronto up in Canada. He has been on the faculty of Vanderbilt University and the University of California at San Francisco. He's the founding director of the Center for Reproductive Biology at Washington State University and University of Idaho, which is the largest reproduction biology center in the world with nearly 100 faculty. Dr. Skinner's current research has demonstrated the ability of environmental response, for example, toxicants, promote the epigenetic transgenerational inheritance, the non-genetic form of inheritance of disease phenotypes to abnormal germline epigenetic programming in gonadal development. Dr. Skinner has over 350 peer-reviewed publications and has given over 350 invited symposia, plenary lectures, and university seminars. Dr. Skinner is the editor-in-chief of the Encyclopedia of Reproduction and the Oxford Publishing Journal Environmental Epigenetics. He has done TED Talks and had documentaries done on his research with BBC Horizon, PBS Nova Scotia, Smithsonian, and France Art. Dr. Skinner has also founded several biotechnology companies. So we couldn't ask for a better expert in terms of the discussion tonight. Dr. Skinner has also, kind of tailored his talk to discussing with public and stakeholders. So without further ado, Dr. Skinner, I'd like to turn it over to you. And first of all, I'd like to say thanks for setting this up on such short notice.
Thank you very much, and I appreciate the invitation. I'm happy to discuss the science. Let me share my screen here. All right, so I'm going to start and talk about a concept that's relatively novel. It's been around for almost 20 years. We identified this back in the early 2000s, and essentially what it is a non-genetic form of inheritance. And it's through a process that doesn't really involve genetics, but it's more what's called epigenetics. And essentially what we'll talk about is this environmentally induced epigenetic transgenerational inheritance, which is a non-genetic form of inheritance. And what I'll talk about this evening is basically the impacts of hatchery conditions on, in this case, steelhead trout populations, but it's pretty much most of the hatchery sort of fish populations. Now, the first half of my talk, I'm going to talk about some of the basic research around this what epigenetics is, this non-genetic form of inheritance and how it works, and how it influences essentially all species from plants to humans, including fish. And so we'll talk about the basics. And then the last half of the talk, I'll talk to you about the specific study we did around the steelhead to discuss that data and move forward. This is going to be presented in a more public manner, so I'm going to summarize things so for basic understanding, and I won't get into the heavy-duty science, but I will show some science slides. But don't worry about that. I'm going to actually summarize things and not get into the specifics, so you'll understand what I'm presenting. Okay, so we'll start with this introduction on epigenetic transgenerational inheritance. So this is the principal paradigm for science today, and it's called genetic determinism. It's been around for more than 120 years. It's what most universities, most classes over the past 100 years have taught. And essentially, this is the basic molecular mechanism that we thought pretty much controlled all of biology. And the concept is that the genetic sequence, the sequence of the DNA, would actually allow certain genes to get expressed. So that's your transcription, what genes are on and off. This would influence the physiology of the cell type and the tissue and eventually the whole organism. And then this creates the biology of the individual or the organism. And when there was a DNA sequence mutation, that would alter expression, alter physiology, and lead potentially to a disease development. So this is the classic paradigm that we've been working with for the last century or so. What I'm going to discuss is there's nothing wrong with this. It's correct, but it's really a small piece of a much bigger story, which we really didn't know about until recently. And I'll discuss that. It turns out that the environment has a really big impact on biology, but vast majority of environmental sort of factors cannot change the DNA sequence. So therefore, it was a little unclear how the environment actually influenced biology. There's a number of different examples of that. The first is if you go anywhere in the world, the same types of diseases are usually present, but the frequency of the diseases change dramatically about where you live. For example, in Japan, there's really high rates of stomach disease and very low rates of prostate disease in the males. In North America, we have really minimal levels of stomach disease, but extremely high levels of prostate disease, to the point where 50% of the males over the age of 50 will have prostate disease. It's the #1 disease in males today. If you move someone from Japan before the age of 5 to North America, as they grow up, they will develop the North American frequency of high prostate and low stomach. So I'm sorry, this can't be due to genetics. It has to be an environmentally induced phenomena based on where they live, okay? So this is one of the first observations that the early epidemiologists in the 1960s sort of observed. The next thing was in the 2000, when we sequenced the genome, we actually started doing genome-wide association studies to identify where mutations may exist that correspond to a disease. And so essentially across the board, they've analyzed pretty much all the different kinds of diseases we know. And for all of them, they've identified the mutations that are associated with that disease. The problem is, if you have 100 people with a specific disease, only 1% of the population has the genetic mutation. So it's an exceedingly low frequency event, and it cannot explain general disease etiology very well. It is part of the disease process, but it's really a very minor low frequency event, not seen in the majority of the patients with the disease. So again, there needed to be something else going on for disease than just DNA sequence or genetics. If you look at the amount of disease in the population, essentially most diseases over the past 25 to 30 years have increased almost tenfold in amounts. It's exceedingly high, and it's still going up. And essentially, there's no genetic process known that could actually increase that speed within the biology of the organism. Genetic mutations don't occur that fast. Again, it's a very slow, low frequency process. So again, something else had to be driving this, and it's likely environmental factors have been doing it. If you take identical twins with essentially the same genetics, as those twins grow up and become adults, they will develop discordant disease, different diseases in each of the twins. If it was all based on genetics, they should get the same diseases, but that's not what we see. Again, the environment of the two twins is apparently driving their disease frequency. There's hundreds, if not thousands, of environmental exposures that are directly linked to disease, but the vast majority of exposures cannot change DNA sequence. So there has to be something else going on in terms of the environmental exposure correlating with the disease. And evolution and rapid evolutionary events is also difficult to explain with genetics. And so, but I won't go into that but very minor in the end. So all of this suggests that there has to be some other molecular process in addition to the genetics. It's not replacing the genetics, it's just adding to it to actually allow things to work. This turns out to be what we call epigenetics. It's distinct from genetics. It's not the same thing. It has nothing to do with DNA sequence. And this epigenetics can actually control how the genome sequence and how the genetics works. It turns genes on and off independently of the DNA sequence. This can then change the expression and physiology and lead to the changes in phenotype or disease. So epigenetics is described as molecular factors and processes that are around the DNA that can regulate what genes are on and off, genome activity, okay? This has to be completely independent of DNA sequence. The sequence does not matter. If there was a sequence dependence, then it would be genetics. So this is completely independent of that. And then it's mitotically stable. So when a cell divides into two daughter cells, not only is the DNA sequence replicated for the daughter cells, but the epigenome marks on the DNA are also replicated to maintain the cell specificity. Since each cell type in your body actually has the same DNA sequence, but a very different epigenetics to give you that cell specificity. So essentially, it's molecular factors and processes that are around the DNA that regulate genome activity independent of DNA sequence, and it's mitotically stable. So right now we know about 5 different forms of epigenetics. The first one that was identified a little more theoretically in the 1970s, mid-70s, was DNA methylation. What they found was there's a methyl group that can get attached to the DNA at a specific base pair called a cytosine residue when it's next to a guanine. And so essentially that was DNA methylation, and it's known that that methylation of the DNA can turn genes on and off by itself. We also now know, based in work in the 90s, that the DNA is wrapped around a core of proteins called histones to create what's called a nucleosome. It's like beads on a string, okay? And so this was, the DNA is actually more of a protein-based sort of nucleosome. And these nucleosome proteins can be chemically modified for acetylations, methylations, lots of different sort of modifications. And that also can turn genes on and off independent DNA sequence. The chromatin structure, where there's tight coils and loops and so forth in the DNA, can also alter gene expression independent of the DNA sequence. So it's a epigenetic factor. There's this concept called non-coding RNAs. These are small RNAs that don't have anything to do with protein translation, but they basically have the ability, these small ones, to bind DNA. They can bind proteins, and they facilitate basically how the DNA works. So non-coding RNAs are one. And the RNA methylation can also change the structure, which is also epigenetics. And so these are the 4 currently known epigenetic marks. I anticipate there's going to be a lot more. We just don't know more about them at this point. So this is the epigenetics. So on the experimental side, in the early 2000s, we did experiments to actually, we use a rat model. We took gestating outbred rat. We exposed various environmental chemicals to this gestating rat around the time of sex determination, when the germ line was developed, the sperm or the egg was developing. So we exposed these gestating rats. Then we actually allowed them to breed and take them to the F1 generation, the offspring. The only exposure was this F0 generation just at a transient period early in fetal development for the fetus. Then we bred this F1 to generate the grand offspring, the F2 generation, and we bred those to generate the grand offspring, okay? And these grand offspring are considered this transgenerational generation, and then essentially the transmission of disease. So the way to think about this is, if you expose an adult, male or female, you expose the F0 generation adult, and you also expose the germ line, the sperm or egg that's in the adult. When there's an offspring, then the F1 generation, that germ line was directly exposed, and so it's basically due to direct exposure toxicity. It's the grandchildren, the F2 generation, that basically are the transgenerational, and they don't have any exposure occasionally. If you expose a gestating female, the F1 generation fetus is exposed, and the germ line inside that fetus that's going to generate the grand offspring is also directly exposed. So you have to get to the great grand offspring to get a generation that doesn't have any exposure. That's called a transgenerational. And so what we've been seeing is, once it's induced, basically it keeps going. And data that I won't go into in detail, basically it's very complex, but all of the epigenetic components contribute to this overall non-genetic inheritance. The non-coding RNAs can direct changes in DNA methylation. The DNA methylation can direct changes in the histone modifications. And so essentially there's the multiple sort of things working together to get this word. I'll talk about DNA methylation because that's what we've looked at, but essentially we now know that they all are working together. So the diseases that we see in a number of the rat models that we've looked at include, for the male, it's basically spermatogenic cell. The testis abnormalities that we see, there's spermatogenic reduction in sort of sperm counts, motility, infertility in the males, and also increased prostate disease. For the females, we see a preeclampsia. We see, in terms of pregnancy abnormalities, we see premature ovarian failure, ovarian polycystic ovarian disease, which is the #1 reproductive disease in females, human females today, female premature pubertal onset, and also parturition defects. And so we see a number of these. For both male and female, we see kidney disease, mammary tumors, behavioral effects in terms of mate preference, anxiety, and stress. And the sum of the frequencies of these are greater than 90% of the population gets this disease. Pubertal abnormalities, obesity, up to 50% of the population, lean phenotypes, and immune abnormalities as well. So greater than 90% of the animals have one or more diseases. And this is a very high frequency event, and it's very reproducible between animals. So I'm just going to show you one example to show you how this works. One of the most recent compounds we've looked at is glyphosate. Glyphosate is the compound that's Roundup ready. It's used to actually in these genetically engineered plant crops like soy and corn and so forth, essentially to get rid of weeds. And so essentially what we found was that as we bred this out to our third generation, like we did the others, we found differential methylation regions, these DMRs, that are present in all three generations. What we found, though, for male obesity, female obesity, parturition abnormalities, ovarian disease, and a whole series of other pathologies. In the first generation, the directly exposed offspring, there was no pathology. So this, due to basic toxicology, has been supported by the toxicologists that basically find glyphosate is relatively safe for direct exposure toxicity. However, if you go out to the grand offspring, or the great grand offspring, the disease goes exceedingly high. And so basically close to 50% of the males are obese, about 45 to 50% are female obesity. Parturition abnormalities are quite high in terms of the females, in terms of birth abnormalities, and basically ovarian disease in the females also go up. And there's several other ones. So essentially what this shows is direct exposure is relatively safe, but there's this thing we call generational toxicology that basically influences the subsequent generations through this epigenetic transgenerational inheritance. And so it's not a genetic-based phenomena, it's more of an epigenetic germline-based phenomena of your sperm or egg that's passing it to the next generation. So this is just one example of a number of close to 20 different exposures that we've done this type of thing. So the way this works is you have an environmental factor that affects a gestating female or an adult male, and essentially affects their germline development, the sperm or the egg, and it changes its DNA methylation. As that individual becomes an adult, the germline now has an altered DNA methylation. It's passed to the next generation. Essentially, the germline changes the embryonic stem cells in the early sort of developing embryo, and they have a different epigenetics and a different transcriptome. Every single cell that comes from that embryo has a different epigenetics and a different transcriptome. It's a cell-specific sort of thing. It changes for all the different cell types, but they all have a shift. Those somatic cells later in life that are sensitive to that shift have an increased disease susceptibility. This individual, as it becomes an adult, through the germline modifications, passes it to the next generation, and thus you have this epigenetic transgenerational non-genetic form of inheritance going forward. Okay, so this is a new form of inheritance, and it's a novel mechanism for our concepts of how disease actually develops. Okay, that it's not due to the genetic mutations, it's due to the changes in the epigenetics of the genome, and it's cell-specific. Okay. And here, just to give you another example of that, if you started out here in the early fetal development, in the early embryo, you have this genetic cascade of gene expression events to eventually get a normal differentiation. You have the same thing going on with the epigenetics. You have this basically cascade of epigenetics, and it's controlling that gene expression. The difference between the genetics, which really can't be influenced by the environment, is the epigenetics early in life from an environmental exposure can dramatically shift the differentiation and epigenetics of the mature cell. This will then have to change the gene expression of those cells, and that shift in of epigenetics and gene expression will increase disease susceptibility or etiology, and also change your phenotypes. Okay, so this is a different way to think about how the developmental biology is regulated through epigenetics. So, as I mentioned, I think there's been now close to 25 or 30 environmental chemicals, toxicants that have been looked at. All of them promote this transgenerational phenomena. Nutrition, both high-fat diets and caloric restriction, will also promote this transgenerational phenomena. And plants, temperature, and drought will do the same thing. Smoking and alcohol will do the same thing. And stress, early in life stress, will also shift the brain development, and this becomes transgenerationally transmitted through the sperm or the egg. This has been shown in plants, flies, worms, fish, birds, rodents, pigs, and humans, and several other species as well. In plants, flies, and worms, this is transmitted hundreds of generations. This is not just a few generations, it goes out hundreds of generations. In fish, birds, rodents, and pigs, and humans, they've taken it out 4 to 5 generations. Just because of the long generational time, it takes longer, but people are starting to get it out farther and farther. So we think it'll still go out hundreds of generations, but the experiments need to be done. So this is this environmentally induced epigenetic transgenerational inheritance. And this, the environment could directly affect that epigenetics. So this is the mechanism on how the environment can change the biology. So I'm just going to give you one example. Here's an exposure of DDT, F0 generations going forward. Okay. Now, if you don't know, DDT was used heavily in the it was identified in the late 40s, was used heavily in the 1950s. In the 1950s, we had the field of toxicology didn't even exist, and the field of epidemiology didn't even exist. It wasn't developed until the 1960s. So in the 50s, they used exceedingly high levels, and in the 60s, they reduced them down, and it was usually banned in North America, at least in the 1970s. And so this was there's really not a female in North America in the 1950s that wasn't exposed to DDT. The offspring then is the F1 generation, and it's been going out, and we are now 3 generations from the 1950s. This is the transgenerational today. In the 1950s, the obesity rate was 5% or less, depending on where you lived in North America. 3 generations later, we're at about 45 to 50% obesity susceptibility in North America. Now, this doesn't promote the disease, it promotes the susceptibility in an individual to develop the disease, okay? So the environmental epigenetics doesn't promote it, it increases that susceptibility, okay? And so this basically, we did the same experiment in rats and showed the same phenomena of about 50% of both males and females that developed obesity or susceptibility. So essentially, could it be that our exposures in the 1950s of our ancestors, great grandparents, essentially caused the susceptibility for obesity today? We need to start thinking about environmentally induced epigenetic transgeneration inheritance as a component of disease etiology today, okay? So that's just one example. Now, what I just described is what your grandparents were exposed to might cause a disease in you, even though you've never seen the exposure, and you're going to pass it on to your grandkids. So that's a pretty doom and gloom phenomena, okay, and concept. So by knowing that science exists and the epigenetics is changing, we're starting to take strategies to see what we could do about it. So here's the glyphosate model that I talked about. And what we did is took individuals with prostate disease, basically kidney disease, obesity, and multiple sort of disease. And essentially looked at epigenetic biomarkers for those diseases in the individuals early in life before the disease was developed, okay? And what we found is we found nice biomarkers. Here's a chromosomal plot with all the different chromosomes in a rat. Here's the size of the chromosomes. The little red arrowheads is where there's a differential methylation site, and the little black boxes are a cluster of these. Essentially, there's no overlap, they're disease specific. So now we can actually early in life potentially diagnose what diseases you might develop later in life, which allows us to use preventative therapeutic and preventative strategies to prevent the disease later in life. And so this will allow us to do preventative medicine. We've started to do that. One of the first places we looked was infertility in the male. About 15% of the population of males worldwide has infertility problems, so it's becoming a major issue. What we found is that in the sperm from these individuals, we found a nice DNA methylation biomarker for the infertility. And essentially here it shows the chromosomes in the human, the size, and again, the differential sites. Here's basically a statistical analysis showing that they're separated. And essentially what we found is a biomarker for infertility and also drug responsiveness for the infertility. So that was our first human analysis to show this is feasible. And more recently, we demonstrated that we compared fathers with an autistic child from fathers without an autistic child. We found a nice biomarker for the susceptibility to pass autism to your offspring. And we had about 805 differential methylation regions. Here's shown on the human chromosomes here and the size. And again, all the red arrowheads are where they're at. A nice separation statistically between the control and the autism transmission. And here's basically showed that a large number of the associated genes were associated with autism previously known. Okay, so this shows we have a biomarker for a male sperm and to diagnose the chances that you may have an autistic child. So again, we could do some preventative medicine based on that. So although it's kind of doom and gloom that this phenomenon is occurring, we can use the science to potentially improve our medical care and our preventative medicine approaches, which we haven't really been able to do in the past. So what I just described was how the environment can affect the epigenetics. It basically can change what genes are on and off, and these changes in the DNA are called epimutations, is what we call them. It alters what genes are on and off in terms of the transcriptome and expression of the genes. It basically changes the physiology, can give you abnormalities and pathologies and phenotypic variation, and it controls the biology and potentially has a role in the disease etiology or what development occurs. Okay, so essentially, this is the summary of the technology that I wanted you to understand, basic science, so that now that we're going to talk about the steelhead project and basically hatchery effects, you'll understand sort of what we're looking at. And these epimutations that go transgenerationally or generationally, these are called ancestral ghosts in your genome in terms of the epimutations. So the paper was published a few months ago, and essentially what we did was we took natural origin wild sort of fish populations, and then we took the hatchery sort of fish populations and compared them in terms of what was going on. Where we did this was at the Matao River in northern state of Washington, and there's a hatchery there called the Winthrop Hatchery, and they're fairly unique, and the hatchery is based on steelhead trout instead of salmon, which more hatcheries are based on the salmon. But essentially, the steelhead trout were here, and they had a native steelhead trout in the river, Matao River population. There's also a dam right below this that basically also had a hatchery called the Wells Dam, and some of the hatchery fish were obtained from this hatchery as well. Okay, so these two were the source for the hatchery fish, and then the natural origin were obtained from the Matao River. Now, the nice thing about the Winthrop hatchery is they use broodstock. They use wild fish broodstock to basically get the offspring and the eggs and the sperm to generate this. And then they basically put those back in the river as they grow up to fry in terms of the hatchery. Okay. And so they use as natural origin fish as they possibly can. Okay. All right. The difference is, though, that these hatchery fish, as they return to the Matao, basically can crossbreed with the wild natural fish. And so we'll talk about that at the end. And so now we wanted to basically look at these fish and look at the hatchery hatchery impacts on the steelhead trout. Here's just basically a picture of these broodstock, these males that were caught as broodstock for the hatchery. Now, it's been well established in the period over the past 10 or 15 years that there's large amounts of impacts of hatchery conditions on the physiology and health of the fish, not only for steelhead, but for just relatively for any kind of trout like the brook trout, a number of salmonids, sort of the Atlantic salmon, Chinook salmon, coho salmon, all of this. So there's decreased fitness due to the hatchery's weird conditions on the individuals compared to the wild. Here's another one where we had steelhead and sockeye, changes in age of spawning, steelhead and coho having lateral lines of morphology, basically in terms of a physiological sort of impact. Steelhead basically increased growth rate and size. Steelhead that basically showed the brain and morphology abnormalities, as well as some salmon. Gene expression changes in body shape, reduced anti-predator activity, delayed hatching, altered migration, reduced egg size. So there's a whole series of these physiological effects that have been shown by a relatively large amount of the literature over the past 10 years or 15 years, showing these hatchery conditions can affect the subsequent fish that are generated. And so essentially, and it's not strictly on steelhead, that's the model we use, but it can basically hit most of these salmonids. Okay, so what we did is we actually took from the natural origin hatchery versus the wild, or versus the hatchery sort of fish that basically had returned, that essentially we found DMRs in the comparisons we made. So again, here's a chromosomal plot with the steelhead. Here's the size of the steel. All the red arrowheads was a differential methylation site. First comparison we obtained was the natural origin versus hatchery, showing this is a number of DMRs that we found. Here's a situation where we actually examined some hatcheries will actually raise their fish with increased nutrition to mature in 1 year. This is in contrast to the normal situation, which it takes a normal fish in the wild about 2 years to mature. And then there's some hatchery conditions where they actually will take the 2 years to mature the fish on a nutrition. So we looked at it, what we call it S1 versus S2. That's a 1 year maturation versus 2 year maturation. So that's something we actually looked at. We found DMRs associated with this. And then we looked at the natural origin fish in comparison to the S1 and S2, and also found, and there's a reasonable overlap between that. So the DMRs were actually generated with all the different comparisons, With all of them, we found good separation or statistics in terms of separation on a principal component analysis. And basically, so that statistically, these were very good differential methylation sites that we found. The CpG density was actually only 1 CpG per 100 base pairs, so it's what we consider a low density, and the size of the DMRs were principally about 1 kilobase in size. And so this demonstrated that basically there was quite a difference between the natural origin and the hatchery sort of fish, and the hatchery sort of conditions of a fast growth versus slow growth would also cause some major effects as well in the epigenetics in the sperm. We did the same thing in the red blood cell, which is a cell that's easily isolated from the fish. The fish have this phenomena where the red blood cells have a nucleus, and so they have DNA. So we did the same sort of thing, and we saw the same phenomena in the red blood cells as well. So if we look at the red blood cells, DMRs, and then sperm, they're very different because they're different cell types, but we had effects in both cell types, as we expect, probably in all the cell types of the fish. When we looked at overlaps, there was minimal overlaps between the sperm and the red blood cells. And there was basically reasonable overlap between basically the natural origins that we looked at and so forth. But for the most part, they were different between the different sort of conditions looked at. What we did is we took the DMRs that had genes that were associated with the DMR. So there's called DMR associated genes. And when we looked at those genes that were associated, the highest number was in the metabolism, transcriptome, signaling. So there's a whole series of different types of genes that were affected, and they were affected in all the comparisons for both sperm and red blood cells in certain sort of pathways. Then if we actually look at these genes in the cell, this is whether they're in the nucleus, whether they're basically in lipids, whether they're sort of signaling sort of things. Here's the sperm, and it shows that it's affecting cell death, angiogenesis, vascularization sort of processes, cell differentiation processes, optic sort of atrophy, and so subcutaneous discoloration of the fish, and basically intellect and brain development. So essentially, these have the capacity to have fairly big impacts on the fish. Same sort of thing with the blood cell, where they have blood abnormalities, those fit fairly well. Again, cell death differentiation. So there were different sort of phenomena going on in the red blood cell, but there were equal sort of effects. So in conclusion for this, essentially what we found was if we have hatchery environmental exposures, we can actually take that out several generations and basically cause these physiological and disease conditions. And essentially, now whether this is, and this is just with the adults, if the exposure of the fish in the hatchery was for a gestating female, then you could get the same thing, but it would take it out. So we've seen this generational sort of thing that needs to be looked at in more detail, but it fits because we're looking at the germ line that's passing something to the next generation. So what I don't want to leave the concept that hatcheries need to be stopped or removed and so forth, because essentially with many of the species we have, partly steelhead in general, the number of steelhead have dropped dramatically in many rivers. And so if we don't maintain the hatchery activities, essentially we're going to lose the fish. So the hatcheries are essential for food production. And in this case, potentially also for the maintenance of the species, we got to keep the hatcheries. So I'm not suggesting we need to remove the hatcheries. But in a recent study that National Marine Fisheries have done out of Seattle, is they actually generated a hatchery with a simulated river. And so they basically have this river sort of process built in where they pump the water to the top of the river and let it go down. So they have this river condition with rocks and trees and everything else. So it's a reasonably simulated river, but they can treat it as a hatchery condition, basically have breeding with broodstock, getting the offspring sort of, and raise the fry and this sort of thing. And so essentially, then when they did this, they looked at the DNA methylation for basically the hatchery conditions versus the simulated river versus the normal. And what they found was the simulated river didn't have all of these epigenetic shifts like the hatchery did. And here's just a different way to look at it on basically in terms of the hatchery showing over here and versus the simulated river. You can see there's a whole series of large numbers of epigenetic changes that were shifted when it was in this simulated river, and we did not see the differences between the wild. And so essentially, what this suggests is if we moved or changed our technology to a more of a simulated river technology, then we could have a population of fish that basically could be maintained without these major epigenetic impacts so that they wouldn't cause these problems with the health and physiology going forward, in addition. Now, in the case of the Matau River, when the hatchery fish can actually go out to the ocean, then return and basically interbreed with the native population, they're essentially passing that epigenetic sort of phenomena to the natural origin fish, such that their offspring have the shift. So over time, that river basically is going to become a more generally epigenetic shifts in which may maintain this thing going forward. Now, in some rivers where there's a dam and they only allow wild fish over the dam, like the Hood River off the Columbia, essentially they can maintain a more wild sort of fish population. So the practices in terms of both the hatcheries and how you manage the interbreeding, if that's now taken into consideration a little bit more because of this phenomena of environmental epigenetics, we might be able to actually have a better hatchery condition to maintain the wild fish population. So this is something, I'm not suggesting we get rid of the hatcheries, but this is something that needs to be considered that's going on now on a molecular level we didn't really know about before. So I don't want to suggest this is all just steelhead or all just a fish thing. We've done a similar type process on Darwin finch in the Galapagos, where we found that basically if the finch live in a rural area, in sort of a wild area, they have certain epigenetics, and if they live in the little city, they basically have different epigenetics and they start to develop different physiological phenotypes. Darwin finch don't migrate. They generally are born and raised within about a 100 yard sort of situation. So they're very close. They don't migrate. This is about 14 miles apart. So they basically have never seen a migration between them. And so what we found was that there was an epigenetic shift in these finch that was associated with these different environmental exposures. That just shows the Darwin finch. And more recently, we did asexual snail. This is a New Zealand mud snail that has a different structure in terms of its shell and ability to actually live in a river or a lake. And so essentially it happens in all species. So my conclusion here is that essentially this is not just geared towards fish, but all species, including humans, are being exposed to the environment and having this shift in their epigenetics. And we need to start thinking about that in terms of the biology. So essentially, we had this environmental exposure that basically changed these epimutations. It changed the gene expression, what genes are on and off, changed the physiology and the phenotype. And then basically when there's this abnormality, you get a disease. If you actually think about this, and it can go forward hundreds of generations, now we have a phenotypic variation that gets passed to all subsequent generations. When natural selection occurs, it actually impacts on this altered phenotypic variation and can dramatically influence the evolution of the species. And so essentially, this is a larger sort of consideration for biology, that this has big impacts on evolution and natural selection as well. It's not just on the individual species at this time. Okay. And these are these ancestral ghosts in the epigenetics. So the main thing is the genetics is very important. Don't want to try to diminish it. In fact, you can't get the epigenetics to work without the genetics. They are actually intertwined. But our older concept that everything was genetic oriented is simply not accurate. The epigenetics has an equally important role in regulating the biology. And it turns out the hatchery conditions, that's what it's doing. It's changing the epigenetics of those hatchery fish, and that's then having these generational impacts later on. So with that, I'll finish, and I'm happy to answer any questions.
First, just want to say thanks, Dr. Skinner. That was mind-blowing, really.
Thank you very much.
Fascinating, amazing science. People are probably still digesting. I've got a couple of questions queued up, and I'm sure they'll start to flow here in just a minute. So the first one, maybe a little bit outside of your your realm. It says, what impact, if any, do you feel CRISPR case 9 could potentially have on defending against transgenerational diseases in wildlife, such as sea lice and salmon, or chronic wasting disease in deer?
Sure. So the CRISPR-Cas is a technology to go in and hit a very specific sequence and potentially modify the DNA sequence structures. You can modify a base pair to get then potentially a modification of how the gene's expressed and so forth, okay? So that's a very interesting high-tech technology. However, what I just showed you was a genome-wide situation. We looked at every single mutation or every single methylation that was different in the entire genome, and there's hundreds, okay? CRISPR-Cas today cannot do more than 1 or 2 or 3 sites at a time. There is no way the CRISPR-Cas could be modified to do a genome-wide effect. And so therefore, it is not going to solve the problem. And it's a very focused, what we call reductionist view, to think that we could take one gene or one site in the genome and impact the overall biology. They're usually, for every physiological event, for every cellular event, there are hundreds to thousands of genes that are involved in that process. We have to stop thinking about simplistic things like reductionism to think about a specific site, and we need to think things better in terms of a system where the whole genome is considered. So I think it's a very interesting technology that needs to be further investigated, but I think we are decades away from its actual application in a systems orientation.
Okay, thanks. Is it likely or possible stress in hatcheries are the main cause of epigenetic modifications?
So I think stress based on the conditions of the hatchery is definitely a major environmental factor. And so I think it will be one. However, an equally important factor will be the nutrition. If you force feed these fish to a degree to get them to mature in a year, it is inevitable the physiology and epigenetics are going to be more dramatically affected. So you have to think about what's the normal situation. And if you sort of try to move away from that normal situation, you're going to find major problems and shifts in the epigenetics. This is why the simulated stream environment is much more of a normal situation. Although the technology is very advanced and it'll take time to actually get implemented, that kind of technology is what will transition the hatcheries to the next generation where we have less impacts on these fish.
Thank you. Very interesting. So the question is, can you share a photo of the simulated river from the Gavry et al. Paper?
I don't have it, and the picture doesn't have it, but you can go to the Seattle Natural Marine Fisheries sort of site, and I believe you can find the photos of that sort of information. So I would look up the paper. It's a Penny Swanson and Gavry sort of paper, and it was published about a year ago. And so, yes, they do have some interesting. I've seen it, and it's very impressive.
Great. Here's a local one. Currently experiencing unusual heat with extreme fire conditions and ash in rivers and streams. Would this current event affect the fish?
So the way to think about it is there's critical times of development that are more responsive than others. The adult fish are going to be relatively resistant to permanent shifts in their epigenetics. As adults, we don't respond very well because all of our epigenetics has been developed. And so some environmental factor may shift things a little bit, but it's not going to change our germline. It's not right. So essentially, we're relatively protected. But early development, so if you're in the fry situation, or early development of the gonads and sex determination, essentially that'll be the time where if you have a prolonged situation of a few months of an exposure, whether it's the fires, whether it's ash, or whether it's the temperature of the water, you might be able to potentially shift the germline early developmental periods to affect the sperm and egg moving forward. And so that's the way to think about it, is when is the best developmental time for those things. And basically, that would be the way to think about it. It's not going to be all the time, and it's not going to be all the fish. It'll be more of a certain early developmental period that's going to be more responsive.
Okay, thanks. If there can be changes in one direction from the environment, can it be reversed eventually by reverting to the unaltered environment?
So evolutionarily, yes. And I suspect some of those things have happened. In other words, where there was this, like during the Ice Age, I suspect, or certain things happened evolutionarily to develop these epigenetically induced phenotypes. As the Ice Age went away, then essentially evolutionarily it was shifted back to actually survive and then not. So this is how species in general, not only man, but all species are able to shift and influence natural selection so they can survive. And basically, it's a very dynamic situation. It's not like it's programmed. And so, but within the individual's lifespan or even a couple generations, I suspect you wouldn't see dramatic effects. It's going to be generational that we actually see it. Okay, but yes, in the classic sort of evolution theory, it's basically natural selection and genetics that's driving the process. Now we know epigenetics is on top of that, and so the environment has a big role. So the environment can actually shift that phenotypic variation, which natural selection acts on. And so essentially that really revolutionizes our way to think about evolution. And so this is the mechanism on how that occurs.
Very cool. With the controlled river system, were the fish milked or did they spawn naturally?
My understanding is the timing of everything was the same. And so it basically didn't sort of shift things, but I wasn't heavily involved in that study. And so you'd have to sort of read that paper a little more detailed and work with the National Marine Fisheries. And they have had much interaction on this. So I think they're more up to speed on that. So I'm not an expert on that.
Okay, great. How were you able to ensure natural spawners were not epigenetically modified? Was there less methylation?
So in the Matao River, where the hatchery fish can return and breed with the wild fish, you can't. And so eventually, because that's been going on since the 60s, I suspect the normal natural or wild fish that was there originally is no longer there. What you have is some sort of hybrid that over time, basically there's been this sort of a crossbreeding between what would have been the wild fish. And that's why in the Matao, they call it a natural origin fish, because it's not exactly a wild fish. It is more wild than the hatchery fish, direct hatchery fish that are taken. And so because we found a pretty big difference in these populations, clearly there is wild sort of characteristics in the natural origin fish, but I wouldn't want to call it a wild fish. Now, if we did the same study on the Hood River hatchery, which has a dam and they try to only allow wild fish across it, it probably would be a better comparison between the hatchery and the wild, because that's not had that interbreeding sort of thing.
Great. This is getting a little bit out of the world of epigenetics. How do you feel about the importance of protecting and remediating natural spawning habitat and of closing harvesting of threatened populations?
And so the last part of that was?
Around closing the harvesting of threatened populations. So more along the management.
So that's a little bit more of a policy thing. And so essentially, it's a difficult question. In other words, for the hatchery production of food, nutrition sort of products, where it's not going to mix with the wild population, we definitely need hatcheries for that. And it's definitely going to be necessary. So this is really why the hatcheries, we can't get rid of them because we need that food production. But today, if we lose the hatcheries, there's certain rivers like the Matao and several others that are right at the level of extinction of the wild fish, that basically we will lose the wild fish. And there's no ability of us to maintain it unless we have the hatchery. Now, I think we should move to a simulated river, a more higher tech sort of hatchery, so we basically have less impacts. And then over time, potentially we can improve that wild population. and so or the natural origin population. But if we don't do that, we will lose those species. It's just inevitable. And so that's why we can't get rid of the hatcheries. It doesn't make any sense, but we can improve their technology to have less of an impact on the epigenetics.
Okay, great. Yeah, that's we have a few questions kind of similarly around those. Is how do you begin to modify the salmon steelhead hatchery design and operations to mitigate these effects? And I think you covered it, but if there's anything else you'd like to add, we have a few questions related to that.
Well, I've actually seen this simulated river, and it's very impressive. It's not like, it's maybe 100 yards at most. And so it's not like it takes miles of sort of thing. It basically is like 100 yards. It's on a nice hill. So there's a gradual sort of river sort of stream going down, and they pump this water and they clean it up going back and forth. And so if you read the paper on that simulated river, you'll see that basically they have some very good ideas. There's some pros and cons about things they did or didn't do. But I think this is the start of that newer technology. And so I don't think you'd actually have to have more, like if they took the Matau River hatchery, they have enough land to actually build one of these things. And essentially it would just mean the investment actually generating the simulated river and a slow transition to actually get rid of the swimming pool type situations. That essentially, I think it's quite feasible to do this. And so if there was an investment to actually do these simulated rivers at the current hatcheries to transition to the new technology, I think that would be the ideal situation to actually mitigate this sort of effects on the epigenetics. So hopefully that answers the questions.
Yeah, that's great. And I think this one kind of ties into put and take versus conservation hatcheries. With breeding only triploid hatchery fish help stop species-wide epigenetic inheritance?
Well, the problem is the triploids aren't going to work to try to maintain the population at all. And so essentially it's great for food production, and it's going to prevent them from reproducing basically if they get loose and so forth like that. But essentially it has its role, certainly in terms of food production, but in terms of trying to maintain the hatchery and the wild population, I think it's a stretch for us to do that, because these are genetically modified essentially, and they're not going to be the long-term solution. If we actually get to the point where, let's say, we lose a species or nearly lose a species, we might bring them in to sort of to maintain something. But I think the better strategy would be to change that to that simulated river type situation, get as wildest population or natural origin population fish as possible, and then try to generate a hatchery that will generate more normal fish. So over time, those become the predominant fish. And I think what we're going to see is their fitness and their reproduction will improve such that we see those numbers starting to go up versus down, which we see in most rivers at this point. So hopefully that answered the question.
Yeah, thanks. And this may tie into the glyphosate stuff you were talking about. Could disease control in hatcheries like parasite S treatments be compounding the other environmental stressors and creating more epigenetic changes?
Absolutely. And I can't say that's the case for sure, but what we've seen in the mammalian models is most any environmental toxicant will actually have effects on that reproduction and the germline sort of epigenetics. So you'd have to do the studies and say, does this sort of treatment impact the epigenetics dramatically? And if so, then you'd want to remove it. If you did it and it didn't affect it that much, then you could basically decide to keep it. But those types of basic research need to be done before we unequivocally just say, let's choose this and put it in, because we don't know if it's worse than not. Just like the glyphosate in the human population, unfortunately, there's no food you can eat today that doesn't have glyphosate in it. Our entire diet, any grain-based diet has glyphosate in it. And so essentially, we as a human population are daily exposed to glyphosate. It may not affect us due to direct exposure, but it's potentially going to affect our grandchildren. So we need to be more cautious about what we're putting in the bar. Now, I can't blame Monsanto and so forth going forward, because they simply didn't know this the 30, 40 years ago, because the science wasn't available. Now we do know it is. And so now we need to start shifting.
Thanks. If the norm is extreme environmental events occurring more frequently, will the long-term survival of these species be jeopardized or accelerated from epigenetic changes?
There has to be evolutionarily a robust population to allow it to actually respond to natural selection and sort of going forward. And so if the numbers drop so dramatically, then unless we get those numbers up, there's just not the number of individuals that will give you that phenotypic variation. And so essentially we are decreasing the chances of maintaining the fish unless we can get the numbers up. And so that's the way you think about it in a general population. Okay. And so we definitely have to have the hatcheries, but we can't have the hatcheries cause more damage than good in terms of fish production if basically we're changing this epigenetics and their long-term sort of fitness. And so that's why I sort of had constantly suggested we can't get rid of the hatcheries, but we have to start moving towards the increased or more advanced technology so we don't cause these effects.
Great. We definitely have a few scientists and researchers here. So this question is, can we state that naturally sized and time releases of enhanced origin juveniles should have fewer potential epigenetic effects than less natural production strategies?
Absolutely. So if you try to basically get a 1-year maturation when the normal period is 2 years, it's inevitable you're going to have more epigenetic effects. So anytime you can move to a more natural situation, both in terms of like a simulated stream environment, this normal nutrition, those types of things are going to be how you improve a better fish with less epigenetic impacts.
Okay. And now this is a little bit more around the hatchery piece. So what type of environmental exposures in the hatchery steelhead are causing the changes? Is it during the spawning process, around, holding anesthetics, that sort of thing, or during the rearing of hatchery juveniles? And then also there's a question if we're able to share some of the papers related to your presentation, and we can work on that offline. Whatever we can send out, we'll send out at a later date.
Oh, sure. The reference for the paper, it's open access. And so you can actually go online and get access to it. There's not a problem. Perfect. And same thing with the simulated river. That's an open access journal as well. So it's all right. So the question, I'm sorry, go ahead, tell me again.
It's around environmental exposures. So is it during the spawning process, or is it as juveniles, or kind of, do you know where it is?
All right. So people traditionally have thought about a specific stage as being the effect. That is generally not the case. You have to think about the entire developmental process from beginning of the germline all the way through to the adult stage. And there's developmental periods at all of these stages where they're getting. So in other words, the epigenetics of the early embryo cell is completely different from the epigenetics of the brain neuron, okay? And it basically progresses halfway through its life until it's mature almost. And so at each stage, you can shift the epigenetics potentially of those cells. Now, it's most sensitive in the early embryo, early fetal period. The next period that is equally important is basically right after birth in the early postnatal period. That fry period is exceedingly important for every tissue that's developing in the fish. And it's dramatically changing its epigenetics. And so if there's an environmental sort of thing, it's going to shift. Now, as fish reaches the adult stage, then they become more resistant to it, just like we are as adults as well. And so essentially, that's the way to think about it. It's the earlier stages that are far more sensitive than the adult stage. Okay.
Great. Using frozen sperm to preserve genetics is becoming more common now in hatcheries. Could we be reintroducing these epigenetic characteristics, and could we test for them before we use them?
Now, it turns out, yes, you could do this, and that would tell you whether that would really be a high-tech sort of screening sort of approach, but you could do that. And if you found that this individual had a lot more epigenetics things than sort of the wild fish, then yes, you should think about that. So you could do a screening, but you'd have to do that genome-wide screening to do that really effectively. If you just had a few epigenetic sites, you may not be able to efficiently look at the differences. And so that would be something to consider. You'd have to do the higher tech technology genome-wide to get the full picture of what that epigenetics was. But theoretically, you could do that. You couldn't really do it on eggs because you can't get enough DNA from the eggs, but the sperm you could easily do that from. And so that would be possible. I mean, I think that basically knowing the origin of the fish would be the first big step in terms of trying to get as most wild population possible going forward. Okay. Hopefully that answers the question.
Yeah. This is just a BC. I think most of the people are from BC. Our steelhead channel is earthen, somewhat stimulating a stream. Is this uncommon for Washington? And is it closer to what you are recommending?
So I'm sorry, what's different between BC and Oregon?
It's the steelhead channel, I guess the spawning channel is earthen. So
correct. The Columbia River has, I don't know how, 8, 10 dams on it. And so essentially that's a pretty big, clearly that transfer through all of those dams and passageways, the temperature of the river has actually been dramatically shifted because of its size. And so yes, it's definitely more of a hurdle for the fish to get through all that into the ocean and so forth. Now, the ocean conditions right now look like they are just as bad, if not the main drivers for a lot of the current health of the fish. And that probably isn't going to change that much between BC and Washington in the Northwest. So but the rivers will be have an effect. And so yes, part of the fitness of these fish is their ability to get through those dams to the ocean, survive, and then return. And I'm sure that it's much more strenuous sort of process in the Northwest than it is in BC, for sure.
Okay, great. Could the combination of triploid hatchery fish for food and natural habitat enhancement for wild fish instead of model rivers hatchery be a better tool to separate genomes back to normal? And I think in BC, we're not as heavily impacted by hydroelectrical development. So we're living in kind of a different space.
Right. So yes, I think if you're in a situation where you're just generating food production, the triploid's fine. However, releasing them in a natural environment into the river and stuff will be problematic in the sense of identifying this. And so essentially there are some problems, but a hatchery that just generates food, you can do that in the current hatchery situation. The problem is you don't want to release those fish to the river because that's going to change their epigenetics and so forth. But for food production, it doesn't matter. It's really trying to maintain this natural origin wild fish, that's where it really matters. And so that's where you'd have to have the simulated environment to get the more normal fish as possible, and then try to stop any interbreeding with the wild fish to essentially keep this and hopefully increase the population of the wild fish. For food production, you could use the current hatchery situations. There's no issues because they're not being released into the river. That's great.
Were any of the epigenetic or transgenerational effects positive from hatcheries?
So it's you have to understand that. Okay. So there's a phenomena, for example, it's called the thrifty phenotype. Okay. And it was initially identified in foxes or wolves, I forget. And then basically now it's been shown in most mammalian species that if you allow the mother to grow up or to grow up and then have offspring in a nutrition caloric restricted environment, okay, when they reproduce, the early fetus is responding to that caloric restriction environment to change its metabolism so that when it's born, it has the possibility to live on a lower caloric diet and survive more efficiently. This is a good example, and most of that is due to epigenetic shifts in the metabolism. In other words, that caloric restriction is changing the epigenetics of the fetus, allowing it to become programmed so that it can live on less calories. Okay, so that's a good thing, and that's a positive thing because it allows their offspring to survive. Okay, now, if you actually put that offspring or its offspring on a normal diet, guess what? the first thing that happens, it develops obesity. It's really, really large obesity disorder situation and reduces its fitness. So an effect physiologically has to be, you have to step back. It's hard to assess, is this a good thing or is this a bad thing? This is an example with the thrifty phenotype. You can consider them both. It's a good for one situation, but it's bad for another situation. And so that's what you can't really pinpoint something that's good or bad. Okay. All we can say is by looking at the last 50 years of research, we see that the hatchery conditions we're using now has decreased the numbers of fish dramatically. And now we know epigenetically, we dramatically shifted the molecular makeup of these fish. And so that's the working point at this point, not whether it's good or bad and so forth. Okay.
Great. Is much known about the mechanism by which environmental factors result in DNA methylation?
Sure. There's a whole, like, for example, so the way to think about this is an interesting question. What do you think makes an embryonic stem cell? Okay. It turns out in an embryonic stem cell, what you have is a DNA methylation erasure. Okay. It doesn't completely go away, but it's like down to 10 or 15% of what it was. Essentially, you remove the DNA methylation. What that does is it creates a totipotent cell so it can turn into anything for all the somatic cells in the body. And so you essentially have this DNA methylation erasure, and then it returns and goes and differentiates into every single cell in the body. Okay. And so that's the way normal development occurs in most every species, is you have these embryonic stem cells, and that's what they do. They have a lower DNA methylation, and then it allows it to develop into what it's supposed to be. Okay. So if you were to manipulate this DNA methylation in a global sense like that, after there's a developmental process, you would lead to a whole bunch of diseases, because that cell would turn around and be able to turn into a tumor or a whole series of things that it would normally turn into. So no, if we tried to influence or change the global methylation, it would be lethal to a degree for the individual, and you get very much more lethal or high phenotypic variations that you couldn't control, like disease and so forth. So because epigenetics is so universal in every single cell type and every single organism, you can't really manipulate it to control what we're doing. At least at this point. Maybe 100 years from now we can, but we can't do it now.
Great. Okay. Well, I have a question for you, and it ties into this using sperm. We have a few situations in BC where we have so few fish returning that we are using some of this kind of banked sperm. So how does that, if we cast back 40 years, we have all of these changes in climate change, and we're kind of bringing, old genetics back. Is there any research, or do you have any idea in terms of what that looks like for these fish going forward?
So if you actually had a really good bank of sperm from 50 years ago and it was stored properly, more than likely you'd have less epigenetic impacts that we see today in today's fish, because it was 50 years ago before we had all this other stuff going on. So there's a good chance, if you know that was from a wild fish and it was stored properly and so forth and viable, that you could actually use that through an ICSI type approach. One sperm gets injected into the egg to actually generate an IVF sort of ICSI sort of fish that would be more normal. And so I think if you'd actually have that sort of resource, then it's probably very viable to actually do this, to try to return the fish to a normal situation. Now, if you use that situation in that simulated river to get more fish, then essentially it might be a controlled environment to get a population to basically reintroduce the normal fish to the river. And so if there was an extinction in a given river, that probably would be a better approach if you have that type of research. So I'd highly encourage you to think about that. And so if that resource is available, that would be, I didn't know it would be a very nice one.
Okay. Any final questions? Dr. Skinner's he's been on the mic for just about an hour and a quarter here. I think you've answered a ton of questions. So
No problem.
I'd like to thank you. Absolutely amazing presentation and research. And I imagine you have other students that are working on this. So love to have you back in another year or two once you
No
have.
problem.
Yeah, that would
No
be great.
problem. Like I said, I really appreciate the opportunity and hope that helps people understand this new science a little better.
Yeah, that's great. So thanks, Dr. Skinner. Thanks, Brian and Josie from the BCWF office. Thank you all for attending. Next month, we're going to be talking about CWD on August 5th from the provincial veterinarian, Kate Nelson. So please tune into that one. And again, for everybody that tuned in, this information is brought to you to be shared with your friends, family, colleagues, and also with your elected officials. We will get the papers that Dr. Skinner referred to earlier and ship them out via our email update. And like to thank you all again and have a great night.
Thank you very much.
Thanks. Good night.